The British Society for Cell Biology, or the BSCB, offers career support to both early career and more experienced cell biologists. The Society awards medals every year to members who have conducted excellent research within their field of cell biology. The winner of the 2026 Raff Medal is Eva Simpson.
The Raff Medal is awarded to an outstanding contribution and a discovery during a PhD thesis.
At the 2026 Spring Annual Conference, Eva was presented with the award for her outstanding PhD research at the University of Nottingham. Eva conducted her work in the Booth Lab, set up by Dr Daniel Booth in 2020 at the university's Biodiscovery Institute. Today, delve into the world of cell biology with Eva Simpson. Congratulations on your win of the Raff Medal, Eva. I was wondering if you could tell me a bit about your PhD research that won you the award.
My PhD focused on chromosome ultrastructure. I was specifically interested in cell division in oocytes because I had a slight background in fertility, so I wanted to investigate this but use some advanced imaging techniques to explore the structure in a little bit more detail. There was two parts to my project. The first was using a technique called 3D clem, which is correlative light electron microscopy, to investigate the ultrastructure of chromosomes. And then the second part was to look at chromosomes in meiosis, specifically in oocytes, which are egg cells, and try and explore how the chromosome periphery, which is a coating around the chromosomes, was affecting ultrastructure at different stages of division.
So, how does 3D clem work, and how did Eva use this to look at meiosis in oocytes?
The reason that we decided to use this for our samples in particular is because the light microscopy allows you to look at specific cellular components or proteins within the cells and find areas of interest or identify rare events. And then when you take it onto electron microscopy, you can get the nanometer precise data of the structure of the chromosomes. With mine, I did light microscopy on the oocytes to identify different stages of division and also see the chromosome periphery surrounding those chromosomes. And then I took those cells, processed them for electron microscopy, and then I could get slices of the entire oocyte using the FibSen, which is a focused ion beam scanning electron microscope. And from that I could create a reconstruction of the entire oocyte itself and get the nanometer precise data about the chromosome structure, and then we could generate those 3D models, and from that we could get geometric information. So this is one of the oocytes that we imaged with the confocal microscope. So we actually used the Z-stack setting to do this so that we could image the entirety of the way through the chromosome region. This is stained with DAPI, so that binds to the nucleic acids inside the cell, and you can see them fluorescing here. By getting a Z-stack of this cell, we can not only identify which stage of division we're in, so if we go to the 3D model, you can see them nicely aligned here and clearly defined along the metaphase plate. So by using this light microscopy, we can identify where in the cell we're actually going to be focusing EM imaging. So in this case, we can transfer this data across to our electron microscope, align our image of our resin-embedded cell so that we can overlay this on the top and actually map the chromosomes to our specific region.
Eva's research identified a novel role for the organiser Ki67. Ki67 staining confirmed the presence of a chromosome periphery in meiotic oocytes. Removal of the periphery was seen in Ki67 knockdowns, which resulted in negative effects on chromosome ultrastructure. This demonstrates the essential role the periphery plays in meiosis. Read more about Eva's research in her publications. So what does all of this mean? And why is this research important? What is meiosis?
Meiosis is a specialised type of cell division that produces gametes, so eggs and sperm. Unlike mitosis, which produces genetically identical offspring, meiosis, it slightly more complicated process. So you're still going to go through the stages like prophase, prometaphase, metaphase, but it will actually go through each of those twice, and at the end of it you will have a haploid cell. This is so that when fertilization occurs and the sperm and the egg combine, they're able to combine that genetic makeup to make a complete cell. It's really important to study meiosis because it's one of the most important biological processes because it underpins sexual reproduction.
So in human oocytes, something like 50% of them display some sort of error, and this is a major contributor to women and couples that suffer miscarriage or have fertility problems. A reason behind this, these issues are still relatively unknown. So the molecular mechanisms that underpin these issues are not very well characterised. One of the goals of our lab is to better understand the mechanisms which underpin aberrant cell division.
So by understanding the chromosome architecture and what each component of the chromosome is doing, we can get an understanding of the entire cell, and I think it's important to have that foundation so that we can build on it, and then over time hopefully that will lead to improvements in patient care.
I wanted to ask a bit about why you chose to study cell biology in the first place. How did you get to where you are today, Eva?
I actually did an undergraduate in biomedical science at the University of Salford because I knew that I wanted to be in a lab. I've always liked science and I knew that I wanted to be hands-on doing work in a lab space. During that time I actually did a placement year at a molecular genetics department, and through that I got to go and work at a fertility clinic. And while I was at the fertility clinic, I honestly just fell in love with it. I thought the work that they did was amazing, and I also got to see all of the science that happened behind it and the impact that that work had.
Eva then went on to study an MSc in medical biotechnology with business management at the University of Warwick.
When I saw this PhD come up with Dan at the University of Nottingham that was focusing on imaging techniques, so I could look a little bit at the technology side of it, but also focusing on meiosis, so it was still linked to the fertility interests that I had. It was the perfect idea for me, to be honest. It was the perfect project.
How does Eva's work fit into the context of the Booth Lab? What are the team in your lab working on, Dan?
So my lab are interested in the structure and dynamic properties of chromosomes. So why? Because this is really important for understanding how cells divide, the mechanisms underpinning cell division events. And this is really important for understanding how this can happen in normal, so in healthy cells, but also in cells where things can go wrong. So cell division errors are linked to a number of human health conditions like proliferation diseases such as cancer, but also related to things like fertility and miscarriage. So by understanding the basic biology, the fundamental biology behind cell division events, we can hopefully work out what causes the diseases and new treatments to target those diseases.
What are the main cell biology techniques you use in your lab?
Yeah, that's a good question. So our lab are a very multidisciplinary lab, so we use lots of different techniques, and what I try to do is have good variety within the lab. So we don't. So what we have is we have a number of students and postdocs that each has their own sort of specialist skills. Every member in the lab has bread and butter skills such as microscopy, tissue culture, molecular biology, biochemistry for doing Western blottings, for example. However, each lab member also has tends to have a specialist skill as well. Perhaps the cell biology tool we've used the most, and the one that I'm most familiar with, is the one that actually Eva is expert in. And so I started working on these sorts of techniques when I was a PhD student myself in Liverpool with Ian Pryor and Steve Royal, and that was setting up multimodal microscopy, better known for our work as CLEM, correlative light electron microscopy.
What was it like having Eva in the lab?
So having Eva in the lab was really great. I was fortunate enough that Eva chose to do a project in my lab. She was my first PhD student in my lab in Nottingham. So Eva turned out to be really dedicated, hardworking, very technically gifted. She's one of the few people that's actually expert in multiple different types of electron microscopy. She's also a lovely personality and a warm and kind person to have in the lab, so she's really easy to get on with, very personable, and was very well liked in the lab. So I guess in summary it was a real pleasure and a privilege to be a supervisor for Eva for those 4 years.
How did you feel when you found out you had won the 2026 Raff Medal?
I was really excited and really honoured to win it. I wasn't expecting to actually win it at all, and it was really nice to know that other people had had a chance to look at the work that I'd done during my PhD and thought that it was good enough to win an award, because I think especially during your PhD it can be quite easy for imposter syndrome to kick in, so it's very nice to have other people look at the work that I've done and think that it is as cool as I think it is, to be honest.
And now that you've finished your PhD, what are you doing at the moment?
Now I actually work as a research fellow focusing on correlative light microscopy at the Nanoscale and Microscale Research Centre, which is always a bit of a mouthful to say. That means that I get to help other people with their projects, so I can advise other people on how they could use correlative light microscopy for their work. I get to do a little bit of my own research as well, and it also means I get to continue with the work that I was previously doing during my PhD.
Along with her incredible work, winning the Raff Medal was made possible by Eva's membership to the British Society for Cell Biology. What made you decide to become a member of the BSCB?
I think it's a really good community for anyone that's interested in cell biology. It gave me a lot of opportunities to attend conferences and events and meet other people that were working in the same area that I was. So it allowed me to go and learn about other people's work that was maybe similar to mine and help me form collaborations, but also see things that people were working on that was completely different to anything that I had looked at before, and gave me an opportunity to think about things in a different way by seeing all of the work that everyone in the cell biology community was doing.
What else can the BSCB offer for PhD cell biologists?
There are loads of great reasons why PhD students should become a member of the BSCB. You get a discounted rate on all of our conferences, and you can apply to travel grants of up to £1,000 to travel to conferences anywhere in the world. Plus, you get to be part of our vibrant ECR community that host events, career development events and online seminars focused specifically at ECR.
To find out more about the opportunities available at the BSCB, and for more information on Eva Simpson and her work, visit the BSCB website.